All right, good morning, everyone. Thank you so much for being here today. We're really thrilled to have you here because today we're going to be talking about our new product that we're launching, which is called Benchling for Lab Automation. A little bit about me, I'm a product marketing manager at Benchling. So today I'm actually going to be dividing my talk into three sections. The first is really talking about Benchling and what is the Benchling platform for those of you who are not familiar with what Benchling is. The second part is going to be more about what's the current situation or a challenge that a lot of you are facing. There's been so much investment and innovation that's happening in hardware, in automation hardware, in analytical technologies, but we believe that informatics is lagging behind. So what do we do about that? And the third section is really going to be talking about our new product, which is the Benchling for Lab Automation, and how Benchling is able to connect seamlessly to your lab instrumentation, both your analytical instrumentation as well as liquid handling. So we are a trusted partner to life sciences leaders. We have hundreds of companies ranging from biologics, cell and gene therapy, all the way to synthetic biology and agriculture. And really our mission is to help our partners really accelerate the pace of their life sciences R&D. So this one question I'm sure a lot of you have is, what is Benchling? We're a unified, and we call it a biointelligent life sciences R&D cloud software platform. And really, the whole idea behind this is that we're one system. Your scientists will be interacting with one system, what allows you to have a unified data model that can centralize, standardize, and also interconnect all of your R&D data in one place. And which means anywhere from your DNA sequences to the physical locations of your samples, to your experimental context or your results. Everything resides in one place. So you're able to really make much faster decisions, gain insights that weren't previously possible before. This is a little bit of a screenshot, a snapshot of how our software looks like, and I'm gonna be showing you a little demo as well. So we call it the Benchling Difference. Our Benchling Life Sciences R&D cloud system, we are a native cloud-based platform, and we really have certain differentiators from the traditional informatics systems out there. So number one is we're purpose-built for biologics R&D. A lot of traditional informatics systems actually were built for small molecule that pivoted then to biologics R&D. However, Benchling was built from the ground up for biologics R&D, so we're easily able to model the complexity of biologics. We're also a fully unified and configurable platform, so you can really track your biologics end to end in real time. What does that mean? You're using the same system when you're discovering, when you're doing target ID, when you're discovering a biologic, all the way when you're taking it to the lead molecule, you're doing cell line development or process development, really getting it ready for manufacturing. So you have one system, and that's completely tied, and you have full kind of traceability. And I'll go into a little bit more detail after. We're also a very flexible platform that's easy to integrate and extend. We understand in your ecosystem you have different systems, different databases in place to do specialized things. We're able to easily integrate with these different databases, and we really pride ourselves in implementing the system in weeks. And you can also tweak it and adapt it as your R&D needs evolve. And the fourth Benchling Difference, as we call it, is really our modern and intuitive user experience. We've really incorporated things that you do normally in your day-to-day life on social media, whether it's something like app mention that you'd be using on LinkedIn or Twitter. We've incorporated a lot of these elements into our software, which creates these unprecedented user adoption and loyalty. Benchling is really divided into three types of applications. The first type is really productivity applications. Second is, we call it sample management or sample tracking. And the third is process management. The productivity applications really consist of our molecular biology and notebook. And what that means is when we say unification, what does that mean for you? So the scientist who's interacting with the notebook can easily register entities directly from the notebook or also track the inventory directly from the notebook. So you don't have to go to multiple systems in order to do that. We also have first-in-class high throughput capabilities in molecular biology, whether that's CRISPR, batch cloning, sequence design, all of that is happening in one platform. And once you've kind of designed your sequence, you can write all the experimental context around it in the notebook, and it's all interlinked. We also have tools available to ensure strict regulatory compliance. So we were built for 21 CFR Part 11 compliance with controlled user access, comprehensive audit trails, as well as electronic signatures. The second type of applications we have are the sample tracking applications, which is the registry and the inventory. So we call ourselves, we have the state-of-the-art registration system because you're easily able to model complex relationships with custom schemas or configurations. What that means for you is you have a mouse, you get the tissue from it, you have the actual hybridoma, the lead, all of that is interlinked, and you can they're clickable links that you can find more details on. We also, as I said, this biointelligent links with full sample lineage that really shows you complete linkage for upstream and downstream entities. We're able to integrate with multiple instrumentation as well, such as liquid handling, such as analytical instrumentation, but also smart fridges and freezers that kind of really easily allows you to run your experiments and centralizes all of that. A third type of applications, we call it the process management applications, which are requests and workflows. And these are really built to power effective collaboration. Number one, as I said, we're a native cloud platform. So sharing between your colleagues, between your partners is very easy. We also have a centralized dashboards that we provide that gives you full visibility into your requests, into your programs, into your status of your programs as well. You can optimize your resource allocation or your productivity. And also, you can now have one platform that's able to kind of unify your discovery and development. So you don't need to be transferring that context, or a lot of times we know things have to be repeated because that context is not fully transferred. Now you have one system that will enable you to do that. So now changing gears a little bit, what's the current situation, and then why did we build our new product? We've seen here, and this is kind of a perfect conference for that, lab automation has really become a critical part of life sciences R&D. I think it was 2 days ago, there was a press release where High-Res Resolution signed a deal with Novo Nordisk to be able to really fully automate their R&D. So we're really seeing this shift in the last 5 years or so, where automation has become a critical part of R&D. And there's several market dynamics at play for that, such as there's emergence of new complex drug modalities, or there's increased competitions, and also desire to reduce cost. So this has contributed to, we call these 3 trends. One is there's increased throughput requirements, right? There's a fail fast, fail early. All companies are trying to kind of scan or screen a lot of molecules with a lot of tests early to improve their probability of success. There's also been increased investment in instrumentation and robotics to really aid that efficiency and accelerate the pace of research. And as a result of that, we're also generating a lot more data than we ever did before. And there's a lot of fragmented data and disparate systems. So that really creates what we call is a bottleneck. So as there's been a lot of investment in automation and laboratory instrumentation, but we feel that companies are still not unable to maximize the benefits from their automation investments. So because lab informatics remain disconnected from these instrumentation. If you look at traditional LIMS systems out there, often they're rigid and hard-coded for a specific process. So if you're running one process and if you're making certain kind of tweaks in it, often it takes weeks or months for you to reconfigure the system. There are specialized solutions, informatics solutions in the market that are siloed, and they're very good for one specific workflow. However, you don't have that end-to-end traceability. And this creates the bottleneck for aggregation and traceability, which impedes the pace of research. And so that's why we feel that we built Benchling for Lab Automation to really help you harness the full potential of your laboratory instrumentation. So Benchling for Lab Automation basically means the seamless and out-of-box integration with liquid handlers and analytical instrumentation. So here, your scientists will be really interfacing with the notebook that kind of runs at the center here. And you're able to generate input files and really parse all the output data that's coming from these instruments. And all the underlying actions that are kind of taking place, that happens automatically. So if you're running certain samples, or you're using certain reagents, all of the information that's in the registry of the inventory will be automatically updated. And you'll have a full entire chain of custody intact when you're using this one system. So what is the product really? What is the product? So one, we're able to generate and run instructions or input file for liquid handlers. This is based on scientist-driven input and parameters, and we're going to see a little demo right after this slide. And also, a big thing is that all of this is using point and click. There is no custom code that's required here, which also gives you the flexibility to be able to modify it as you see fit. We're able to ingest the output files. So again, the configuration is easy. As I said, the whole benefit of having the centralized unified system is that even though there's one API endpoint that connects these instrumentation, you're able to have all the actions underlying, such as in the registry or the inventory or the requests, happening automatically. So, which could lead to the creation of results in your notebook. It could, if you are creating new samples, you're changing that in the registry. You're updating samples or reagents that you used in these experiments in the inventory without any manual intervention. Traditional LIMS systems often, right, or you invest in a piece of hardware to accelerate your research, oftentimes scientists still had to manually go in and input data into multiple places. Now we kind of take care of that with our unified system. So wanted to show you our product demo. And really here we want to show that Benchling seamless integration, it's with the Hamilton robot system for a typical hybridoma workflow. So we start after the fusion is complete. And then we're going to be plating the fusion into 96-well plates to ensure that there's single cells in a plate. Once that's complete, you will see how we're creating the input file for the robot to execute that and then parse the output from there. Once that's done, we'll let the cells grow, and then we'll be pulling the Supernatants. And again, you're going to see how Benchling will enable that with the input file creation as well as the parsing the output, and then the data from there once these Supernatants are screened. So let me just quickly play that. All right, so here is what I was talking about. Your fusion is complete. You're going to be plating that. Here you're seeing that there's two different fusions being used. We're creating an input file from it. So which is what I was saying. The input file is really created with scientist-driven input parameters. Here we're using one fusion that's going to be plated in 8 plates to ensure that single cell is deposited into the 96-well plate. And the second fusion will be plated into 2 plates. We're going to be adding about 200 microliters in there. So that's the input file. So let me just pause here for 2 seconds. This is the input file that you're going to be providing to the liquid handling robot. So in this case, it contains what the sample ID is, what's your source tube, what's your destination tube, destination well, and what the transfer volume is. Your liquid handling robot will take this information and then execute the method. And once it does that, we're going to be ingesting the output. So here you can see the way we're doing it, we're actually doing it in the user interface. And it's very, very easy to do. However, if you want that automatically done, we could build a custom integration for that as well. So once we process the output file, this is where the parsing begins. So in this case, what you're seeing is these are the different plates that are created, and these are all, they're entity links. And in the next example, I'll show you how it's all connected. Now we've plated that in hat media. Now we're going to let them grow and pull the Supernatants for screening. So again, the input file is similar. So in this case, we're providing what your sample input is. What is your destination? In this case, there are plates we're going to be putting that in, 96 well plates. And also, what I want to really stress here is that if your robot has a barcode scanner, you don't have to create these plates. They'll be automatically registered, but you could also pre-register them if that's not the case. And again, so you have that complete sample chain of custody intact throughout the workflow. In this case, we're going to be pulling about 100 microliters of Supernatants. We're generating the input file. Oops, sorry. Oh no. So here it's really showing you what you pulled your Supernatants and which well it's being deposited from. And as you're creating these new Supernatants from Hybridoma, you're actually registering new entities automatically. So you can see that the scientists actually didn't have to go and register them. This was done automatically. And if you see the output here, this is what I was talking about, complete lineage. So if I click on the Supernatant, and if I click on one of the wells, you can clearly see on the left what was the disease model, what's the mouse it came from, what was the fusion, and what was the hybridoma. So as you start kind of building up, whether it's your lead, you're doing antibody engineering, you're doing affinity maturation, you will see these entity links, and you can actually go back and kind of track the entire lineage. In this case, then we've pulled the Supernatants, and we're actually recording titer data against it. So similarly here, this will show you how we're going to have a results table. Give me one second. Here we're going to be plotting as well. So sample ID against the titer. And this is really what we're saying. Once you've tested your antibody, you tested your Supernatants for antibody production, and you want to hit pick. What do you want to take for expansion? You can do that. And this is again showing you that you have full visibility even with the titer data, and you can kind of track all the way back. So everything is being recorded in the registry, and you could, as I said, you're never losing any information. It's not happening in disparate systems. It's all unified in one platform. Here's a graph where we just plotted the titer values, and we have determined what the threshold is. And so we've picked the hybridomas that we want to carry forward. And we've created the list there. Again, we're going to consolidate that in one plate. Generating an input file for that. And then we'll upload the output. So now what we've done is we've chosen about 96 hybridomas that we want to take forward and consolidated that in one plate. And then we're going to expand that further. Perfect. All right. So the question is, why choose Benchling for lab automation, right? I keep saying this over and over again. We're unified with other Benchling applications, so you don't have to enter data into multiple places. They're all unified. You put it in one place, and it's available into all other applications as well. We also provide this rich sample and process intelligence. In addition to having that full lineage, you also have entire experimental context behind it, because you can access the notebook entries for each one of them and see the process as well as sample lineage, which is important what we were talking about, because you have one unified system now. You can transfer knowledge very easily between groups or between partners. And the third is really what we pride ourselves is that we built the system on a very codeless kind of configuration. That's important for you because you want systems that are flexible, especially as we're moving into new modalities and your process is constantly evolving. You don't want to be waiting on a consultant or the company to come in weeks or a month's time where you're kind of waiting for that to be changed. Here, you have the power. The customer admins can change that in a matter of few minutes. And we're also vendor agnostic. So whichever liquid handling robot you're using or analytical instrumentation, we have APIs available to be able to integrate with that seamlessly. And to help this kind of integration further, we've also partnered with leading automation vendors such as TCAN, Hamilton, and PAA to really demonstrate our rapid integration abilities. And we hope to expand this further as we move forward. And so, as you said, the Benchling Difference and why we've become the indispensable tool for next generation life sciences R&D companies. We're really solving the problem where informatics was lagging behind. We're unifying hardware and informatics, so really to help you accelerate your pace of research, whether that's with end-to-end traceability, accelerating your speed to milestone, and also more importantly, having access to data-driven insights. Because all your data rests in one place, you're able to answer questions or ask questions you were not able to before. We have customers who are running a lot of queries and creating these dashboards and trying to optimize their process, optimize their resource allocation, and really take out bottlenecks to accelerate their pace of research. We'd love you to come by our booth, which is booth 151. Please come to learn more, and thank you so much for being here.